This video is going to explain PCR. It's a chain reaction made by an enzyme called polymerase. What this reaction does is basically transforming a little piece of DNA into lots of DNA. You might ask, why would I ever want to do a PCR? Well, there are many reasons, but I'm sure you've seen that movies or TV show where the criminal leaves hair, blood, or saliva at the crime scene, and the CSI uses it to find out who's the suspect. Well, you shouldn't trust everything you see in these movies, but in that little piece of hair, saliva, or blood, there's a tiny amount of DNA. In order to work with it, lots of copies need to be made. So the CSI performs a PCR and copies the DNA over and over to confirm the suspect's identity. What I just described is called DNA fingerprinting, but there are many other uses for PCR: to detect harmful viruses or bacteria in your blood, for paternity testing, to detect gene expression, and many other applications. The next question is, how do we carry out a PCR? What materials do we need to copy a piece of DNA? We need some DNA, of course. In our example, the DNA that we found on the criminal's hair follicle. We refer to this DNA as template. If we already have our template DNA, we'll need a polymerase. This is the enzyme that makes DNA. When it sees a strand of DNA, it goes there and makes a complementary strand. We also need to provide the polymerase the building blocks of DNA. They are called deoxyribonucleotides, DNTPs. It's important to include magnesium because the polymerase needs to work. Lastly, keep in mind that the polymerase cannot make a whole strand by itself. It can only add to an existing strand. So we'll need a primer. Primer is a very small piece of DNA that will bind to the template at a specific place and give polymerase a chance to start working. Before we move on to doing a PCR, here's a fun fact about the polymerase that we use. The polymerase we use actually comes from a type of bacteria called Thermophilus aquaticus, or Taq Polymerase. These bacteria live in hot springs and their polymerase is thermostable. It means it can resist high temperatures. You'll understand why this is important in a minute. So let's get into the reaction. We will have in our mix our DNA template, polymerase, DNTPs, and primers. We use a machine called thermocycler. What it does is simple: heat. The machine will raise and decrease the temperature in cycles. Every cycle has 3 steps: denaturation, annealing, and elongation. First, the machine heats at 95 degrees and makes sure all the DNA is denatured. It means that the 2 strands are separated. Later, the temperature decreases to allow the primer to attach to the template. This temperature varies depending on the primer and other factors. Lastly, the temperature rises again, but this time it goes to 78 degrees, the optimal temperature for polymerase to work. Then, the enzyme helps the DNTPs to make one new strand. This was one cycle. Usually it takes about 30 cycles to make enough DNA copies to use in an experiment. The other cycles will repeat the 3 steps, and a huge number of copies of the designated region will be amplified. This is PCR. One last thing you need to know. Note that some experiments will amplify large regions or the whole genome. Some will amplify only small regions corresponding to certain genes. The reaction will amplify only the region between the 2 primers that you designed. The target sequence is called amplicon. It is important to note that your amplicon is not even present in the first cycles. This is not your amplicon, and this is not your amplicon. But look at the second cycle. This is your amplicon. Right now, there are more unspecific strands than amplicons, but as the cycle goes, the amplicons would outnumber the unspecific sequences. Now that you understand PCR, we made you another video actually showing you how to perform a PCR hands-on. See you soon.